arteriosclerosis, hardening of the arteries, and a heart attack. And that occurs, depending on how severe the case is, your first heart attack can occur between the ages of 20 and 30, or between the ages of 40 and 50. Okay, so this is a picture of exocytosis, where there's some substance, perhaps a digestive enzyme, that we've created a whole bunch of inside the cell, and we need to move it into the lumen of our stomach or into our intestinal tract. So we create a vesicle around them, move it to the surface of the cell, and then they're released out into the lumen. And this is a transmission electron micrograph showing that same process. All these little things were things created by the cell, and they're being expelled out of the cell. So here you see the plasma membrane, and this is the membrane around that vesicle. This is receptor-mediated endocytosis. You can see there are these receptor proteins, and once these 5 have bound to the target molecule, then we begin creating that vesicle around them and bringing that vesicle into the cell. And this is a transmission electron micrograph of that same process. Okay, so that's kind of like wholesale movement of substances into or out of the cell. We can also be rather selective in what we're moving into and out of the cell, or perhaps said more accurately, we can move very small things into and out of the cell. And that's accomplished by protein pumps, where we are pumping specific atoms or ions into the cell and out of the cell. And that's best exemplified by the sodium-potassium pump. With the sodium-potassium pump, what happens is we generate what's called an electrochemical gradient. Now that's a 2-part term, electro and chemical gradient. So there is some electrical component to this gradient, and there is a chemical component to this gradient. And I'm gonna treat both of those independently. Now, the sodium-potassium pump is active in our neurons. Our neurons want to keep a specific concentration of the sodium and the potassium ions inside and outside of the cell. What they want is to have a high concentration of potassium in the cell and a high concentration of sodium outside the cell. And so, every time our cells come in contact with, our neurons come in contact with the sodium inside their axons or perhaps their dendrites, then this pump grabs them and shoots them out. And every time there's a potassium outside the cell, it grabs it and pulls it in. And it does it like this. There is a carrier protein that you'll notice has 5 active sites. There are 3 on the left here and 2 on the right. Now, when opened inside the cell, it's sensitive only to sodium ions. Sodium ions carry a positive charge. Now, when it's open in here, these 3 sodium ions can bind to each of those 3 sites. When that happens, then we expend a little bit of energy. A molecule called ATP, which has 3 phosphates on it, comes along and drops its terminal phosphate off on that protein pump. When that happens, the protein changes shape, and these active sites change their sensitivity. So this phosphate is now attached to our pump. It's open on the outside, and these sodiums are going to be released outside the cell because those active sites are no longer sensitive to sodium. That protein has changed its shape, and it's no longer sensitive to that pump, to that ion. However, it becomes sensitive to this triangular potassium ion. So 2 of those potassium ions come in here and interact with those active sites. When the sodium has been expelled, and when the potassium has been attached to, that phosphate drops off, and the protein resumes its original conformation. And it can now pick up more sodiums from inside the cell and pump them outside the cell. So this sodium-potassium pump maintains 2 concentrations of ions, right? A high concentration of potassium inside the cell and a high concentration of sodium outside the cell. But there's also an electrical gradient. And the electrical gradient is somewhere between 50 and 200 negative millivolts. Now we create a negative charge inside the cell relative to outside the cell. But it's interesting, right? Because we're doing that by moving 2 positively charged ions around. We're moving positively charged potassium in the cell and positively charged sodium out of the cell. But the way that we generate that electrical differential is by moving fewer positive charges into the cell than we are moving out of the cell. So really, it's inherently a positive charge. It's just less positive than outside the cell. Now that's the resting state of a neuron. It's maintaining this electrochemical gradient so that when it receives a stimulus, it's able to open up a protein channel and allow sodium into the cell and potassium out of the cell, and which equalizes the electrical differential across the membrane. That's signal transmission. So when a neuron receives a signal, that gate opens up and those concentrations go away. That's a signal that's transmitted down the length of an axon. So the sodium-potassium pump, just to sum this up, is important because it maintains an electrochemical gradient that stimulates or facilitates neural responses and neural communication. So this is the last of chapter 4, and what I'm going to do now is just introduce chapter 5.